So I'm Simon Graham. I'm an MRI physicist and neuroimaging scientist at Sunnybrook Research Institute. And Alison Sekuler is the Vice President of Research at Baycrest Health Sciences. I'm going to talk at the beginning, and Alison is going to follow up at the end. So this project for the both of us started a few months ago, and it was something that the first thing that I was, there we go. Sorry about that. I'm getting confused with my slides. Let's see if I, yeah. So a couple of months ago, the first way that I got interested in COVID-19 and its impact on the brain was through a medRxiv paper from some people in Wuhan, China, that were investigating a couple of hundred of COVID-19 patients that had been hospitalized. And what they found was that there was a significant proportion of people that had had some sort of nervous system complaint associated with their time in the hospital. So this was something on the order of, over all of the various symptoms, something like 30 to 40%, depending on whether the patients were in a severe state or in a moderate state while being hospitalized. And you can see some of the symptoms are things like headache, impaired consciousness. There's some people that had stroke. And then there are some peripheral nervous systems symptoms as well, things like impaired taste of sense of taste and smell. And so the question was for us, what was potentially going on in the brain, and was this a major problem that, given that it's a global pandemic, how many people in Canada are really going to be faced with these issues as they become infected? So the research has been really evolving rapidly. This is another preprint of an autopsy study which shows something about the mechanisms of how COVID-19 could enter the brain. So if you can see here on the left of the slide, it's in the oral cavity. It could get access to the trigeminal nerves and the trigeminal ganglion, or it could get into the brain, into the olfactory bulb from nerves in the nasal cavity. And it could make its way down into the brainstem. And you can see from these autopsy plots that a fraction of people were found to have the virus actually in these areas located in the cartoon above. So this, although it's a preprint, is pretty definitive evidence that in bad scenarios, it is often the case that COVID-19 can enter the brain. So another potential route of entry is through the vasculature. And so this slide is just indicating how the virus, once it gets disseminated into the bloodstream, could target certain organs in particular, such as the intestine, the kidneys, the lungs, the heart, and in particular, the brain. And so, there are some examples of this happening. People are seeing case reports in terms of pretty worrisome signs in certain patients. So this is an example of hemorrhagic lesions that were developing in a patient that had COVID-19. And you can see that the areas are sort of the frontotemporal and thalamic areas. And there's now a number of these kinds of case reports in the literature of patients that have suffered similar sorts of things. In very recent weeks, there's been a number of these papers that have come out. Here's another example of some images where you can see subtle effects at the cortex in the areas where the arrows are located. And this is a particular patient series where most of the people had come off ventilators. So there's a bit of a confound there, because people that have ventilators or put on ventilators, the act of doing that is quite rough on the brain as well. So it's kind of hard to disentangle the virus effect from the ventilator effect. There are now some review articles as well on these case reports. Some people saying that, like in the top review here, that this is an important effect that needs to be considered within the population. And then there's also some counter reviews saying that most of these studies that have seen effects so far in hospitals are not very well designed, and they could be just because of the ventilator effect. But I think actually the autopsy study is pretty definitive. But based on these reviews and what we were seeing in the literature, we decided to develop our own study, which we've called NeuroCOVID-19. It has two components to it. There's a clinical research component, which involves basically taking people after they are no longer infectious and studying them in two groups. So there's a group that are admitted to the hospital, or were in the hospital, and a group that were self-isolated. And then we have a control group who have gone through some sort of symptoms like shortness of breath or cold or flu-like symptoms, but have actually tested negative for COVID-19. And then our plan was to do two sets of assessments after they are out of their infectious period, separated by 6 months. And we were going to do a series of assessments that involve MRI, sensation, cognition, and electrophysiology. And at Sunnybrook, there's a range of collaborators across multiple disciplines. And we also have been doing this in partnership with the folks at the Rotman Research Institute and Allison. So I should also say that there's a bit of a preclinical research component, which is going to be done on hamsters, looking at the effect of infectious dose. So if the dose is increasing, does that mean that there's worse of a brain effect? And also a dietary intervention. So we're going to look at animals with normal diet, and then a diet that's bad for them, basically cafeteria diet, which destroys, I shouldn't say destroys, but causes inflammation and perhaps leakiness in their blood-brain barrier, which would be hypothesized to enable more virus into the brain. And then we're going to study the animals with 2-photon microscopy, either at the cortex or an optically cleared tissue, and looking at the leakage of a compound called dextran out of the vessels to see whether or not we can see virus outside of these vessels with additional histopathology to look for the virus itself. So this is just an example of some initial imaging findings that we've seen. The human imaging part of the study is up and running, and we've now looked at a handful of subjects, and you can see on these anatomical images some small dots, which are likely some sort of lesion induced by COVID-19. Obviously, we don't have the baseline imaging for these people, but we do have anecdotal reports, in particular for this individual, that he just wasn't behaving quite the way he was prior to having become infected. So we'll be looking at that more with our imaging protocol, which is anatomical and functional imaging with a range of different imaging sequences, and then seeing whether or not that agrees with behavioral and electrophysiological data that we've collected. So that's all. Those are all the slides I have. I think Allison's going to take over from here and talk a little bit about the behavioral assessment, sensory assessment, and the electrophysiology, and sort of the wider ramifications of the study. Allison?
Thanks for such a great opening, Simon, and thanks to everyone for being here today to share the time with us. So as Simon mentioned, this particular individual was having some sort of behavioral symptoms as well, but we wanted to do more than just ask people, how do you feel, or you know, do you feel like your perception or your cognition isn't quite up to snuff? So we have a whole protocol involved with sensory testing, including some that's done on site and some that's done in follow-up virtually. And some of it is looking at some of the things that Simon's already talked about, for example, smell and taste, how that's changing. And so doing it in a rigorous way, so having an actual objective sort of test, as opposed to just asking people to subjectively tell us what they're thinking. We also are doing a whole range of cognitive tests and vision tests. And you might say, well, that's weird. Why would you care about vision? I'm a vision scientist by training, and so I'm biased. But we have known for a long time that because some of the neurodegeneration that we see in aging, in Alzheimer's and related dimensions and so on, you can sort of tell that it's coming before there are the more traditional kinds of impacts on memory and attention by changes in the visual processing. We view the ability to perceive different kinds of perceptual tasks, whether it's in vision or audition, as a sort of a screening tool or a precursor for telling us that there may be some other more challenging issues that the person's going to face in terms of, say, memory and attention. And the other reason that we're testing different kinds of vision tests is because, as Simon mentioned, some of the evidence is suggesting that there may be differences in frontotemporal areas or in thalamic areas, and different kinds of visual tests we think are tapping into different parts of processing in the brain. And so by testing to see how somebody does on really low level vision tests like grading acuity, for example, versus a contour integration task versus an oddball task, for example, we're testing at differential levels of brain processing. And so the idea is that that would help us understand in general how COVID may be impacting your brain function, but then also we can link it back to these kinds of images that Simon's showing here to see, in the case where someone has a particular kind of a lesion, does that line up with where we would expect the behavioral or the sensory deficit to be? At the same time that we're doing those kinds of tests, we're also measuring EEG. And all of this, as you can imagine, is quite a challenge in the time of COVID, because normally when we do EEG, we're using in our lab a 128 channel or a 64 or 256 channel system, which, to keep that clean and to keep it sanitized in between patients right now is not something that we're able to do. Fortunately, we have this kind of a device, which is called the Meuse, which is a portable EEG system, and it just works like a headband. You just put it right on your head, and it ends up, it has only 4 electrodes. It's got 2 in the front and then 2 that end up behind the ears. And a grounding electrode as well, which you can see some of the electrodes here. And then these bits here are actually electrodes as well, embedded in there. And the advantage of that kind of system is it's highly portable, because we're actually not able to run people right now in the Rotman Research Institute, because we have long-term care home on site, and we obviously have a lot of precautions that we're taking to keep our residents safe. We are doing all of the experimentation at Sunnybrook. So it wasn't feasible for us to bring our whole system over, figure out how to get it cleaned and so on. So we had already been doing quite a lot of work with the Meuse, which is in partnership with a company called Interaxon, which has developed the Meuse. And we were able to show, as have other people across Canada and other places, that you can get quite good EEG signals, including ERPs, including standard time frequency sorts of analyses from this really simple system, and it's very, very easy to keep clean. So it's portable and easy to clean, and so we're making as much use of it as we can right now. The idea there is that some of the previous work that we've done at Baycrest has shown that the combination of behavioral responses and EEG responses to a variety of different kinds of tasks, and this is work that's being done in other places as well, but we've been able to show that some of the specific tests that we're using in the NeuroCOVID study are associated with mild cognitive impairment and neurodegeneration. So even though it's a very basic kind of a vision task or a very basic cognitive task, when you combine the behavior with the EEG results, you learn a lot more, and it may be a marker, we think, of coming decline. Is what's the thinking. And then that will help us when we do the 6-month follow-up to sort of see if it's able to predict who's more at risk for different kinds of problems than others. The other thing that we're going to be doing is a full assessment of emotional status. So that's being done virtually. So we're doing the remote testing for that using the NIH toolkit, as well as some other standard cognitive measures and so on and so forth. Also taking baseline EEG so we can check and see if things like alpha waves are changing, because we know that those are also related to your ability to focus your attention and so on. So we're casting our net fairly wide. I don't think it qualifies as a fishing expedition, because we actually have reasons for having included every single test that we are including. And it's based in part on the emerging literature that was being developed as we were working together, the Baycrest and the Sunnybrook group, to develop the protocol in the first place. And since then, we've also been hearing more and more evidence that I think supports the choices that we made at the very beginning. The last thing I want to mention, though, is that although this started as a Baycrest and Sunnybrook project together, it has expanded since then. So Simon and I have been working quite hard to bring in partners from across the country, and we're really happy to be sharing this with everybody on the CanCOVID site, because we think that's really one of the best ways to get people engaged across Canada. And we have people who are interested in being part of this study from the experimenter perspective in British Columbia, in Alberta, in Quebec, in other parts of Ontario besides just Toronto. We're looking for partners across the country because we think that this is something that's really important, and it's something that no one location will have the facilities to be able to bring in enough people to really get enough data in a rapid enough time cycle that we can make an impact. Because as Simon was talking about the different potential ways that COVID may be impacting the brain, you know, on the one hand, yes, we're really concerned that if what's happening is that COVID is sort of acting like a series of little mini strokes over time, or a series of concussions over time, and so there could be long lasting impacts. There's a long-term impact to worry about, but there are also maybe short-term ramifications of our results. So if we can find out that the way that it's entering the brain is impacting mostly one part of the brain, and then that part of the brain is actually impacting the way that you control your breathing, for example, well, then that changes the way that you might want to treat breathing problems, right? Or it may show that certain sorts of visual deficits are a precursor for somebody developing a much more severe case, in which case you can have a different kind of a screening tool. So although you might think that what we're doing is not as urgent in some ways as someone who's trying to create, you know, a vaccine or doing the serology testing and so on, there could potentially be quite short-term impacts that are really significant for the treatment of COVID in the short term and in the next phase, which we're likely to end up seeing at some time in the coming months as well. So with that in mind, we also want to be sure that we're testing as broadly as possible. Right now, we have the test site up at Sunnybrook. And as I said, we have several other test sites identified, but we're also trying to make sure that we can be testing remote locations, Indigenous communities, communities of color, people who don't have access to the sort of the main testing centers in the Torontos and the Vancouvers and the Montreals of Canada. So to that end, we're trying to put together another proposal that will help us create a mobile testing unit so we can sort of take NeuroCOVID on the road and be doing at least a sort of a sparse version of NeuroCOVID to get a much bigger sample. So Simon and I and all of our collaborators at Sunnybrook and at Baycrest and across Canada are open for as many different collaborators as possible. One of the things to keep in mind is that we recognize not every site is going to be able to do everything exactly the same. So, you know, some of the sorts of facilities that they have at Sunnybrook for testing may not be available at others. And on the other hand, you know, there's no 7T site at Sunnybrook, but there is at Western, right? So it could be that if we bring on different sites, the core of what we're testing may be the same, but different sites may be adding in their own specific elements to address, you know, more targeted questions that their particular groups are interested in. And I think the last thing I just want to say that is that recently, as some of you may know, there's been a concerted effort to try to bring neuroscientists together across the country, and you know, through support from CIHR, for example, there's been, you know, work behind the scenes for several years. It's moving forward, and I think this is a great opportunity for us to be sort of binding the neuroscience community together and making a real impact in a critical situation. So I think we just want to open it up for questions at this point.
Yes, thank you. That was absolutely fascinating. You've already got some questions popping up. Why did you choose post-infectious and 6 months later as the 2 time points? Specifically, why choose the 6-month time scale, and why not more time points as well?
I can start, Simon, you can join in. The 6-month came because initially we were working, you know, in the context of trying to make sure we could get data very, very quickly to make the biggest possible impact. And it's just not feasible for us to be with the fun, honestly, with some of the funding opportunities that were coming out, if you didn't have something that was planned to be done within a year, you weren't even eligible to be applying. The plan really would be to start with 6 months and then follow up later, because we do know with SARS, for example, there are examples of people, at least anecdotally, showing years later still having cognitive deficits after having SARS infection. So this is just the initial starting point, but we are hoping to be able to do later time points. On the why is it post-infection, again, that was more of a practical reason. We would love to be able to expand it and be testing people during infection. But from a practical perspective, just to be able to work out the logistics of being able to test people even post-infection was quite difficult in this time period. So Simon, I don't know if you want to speak more to any of those points.
I think maybe just for brevity, I won't. I thought you answered that pretty well. I mean, there's more I could say, but maybe we'll move on to some other questions.
Okay, great. You mentioned that patients who had been on a ventilator presented certain confounding factors. How are you working around this with NeuroCOVID?
Well, that's why we're, Allison, I'm going to take this on.
Yeah, you take it.
So that's why we have two different groups of COVID positive, or two groups of people that were previously infected by COVID. So the people that are hospitalized and not hospitalized. So the people that are hospitalized, within that group, there's going to be some that go in the ICU and go in a ventilator, some that don't. We do expect that that group, though, overall is going to have more of a brain effect than the group that stays home. The other thing that we've got in our imaging protocol is that we have some lung imaging. So we're hoping that what we can do is, through the combination of the brain imaging, the stratification of people, the time points, and the lung imaging, is that we can tease out some of these confounding factors in certain people.
Great, thank you. And Avril asks, are you testing motor impairment or function?
Yeah, right now we're not testing it in any sort of rigorous way. We are quite concerned about motor impairment and function. The problem we faced is that there were so many different elements that we needed to bring in. And with the scanning protocol, plus the cognitive and sensory protocol, plus the additional demographic information, it already is a really long protocol. And some of the systems that we've been using to test motor function, for example, at the work that we do in aging and neurodegeneration at Baycrest, require, I mean, I know there's other ways of doing it, but we have, the big mats that measure how people are stepping and pressure points and balance and all of that kind of thing. And again, because we weren't able to test in our site, it was a little bit tricky to be able to figure out how to do it. So there are more basic approaches that we could take if people are interested in joining in. We're welcome to, we're interested in seeing what would be of most interest to the community in terms of expanding the protocol. And, because we're still early days, so I think we still have time to add things and subtract things as needed. And so we're really quite open to it. I think, depending on exactly where the damage is in the brain, you might actually expect there to be significant impacts in motor, and especially thinking about some of the non-traditional kinds of symptoms that you see in older adults in particular. They may not have a fever, they may not have a cold. The first symptom they get is that they fall down. So I think it's increasingly becoming clear that there may well be motor impacts or balance or something to that effect. And so it's definitely, we're open to it, but it's just not really being included right now.
So Avril, step up. Julia asks, will you also have a control group for prolonged hospital stays that are not COVID related?
Simon,
let's alternate. You can take this one.
Oh, excellent. So we hadn't really considered that at this point. So I think I understand the concern there that, a prolonged hospital stay, somebody's going to be weak, they've been bedridden, that's going to have an effect presumably on their brain as well. So I don't know, actually, Allison, I'll bounce that back to you. What do you think? Do we need another control group?
We could, if we end up with enough people in our COVID recovered group who had prolonged hospital stays, then we would want to have that additional control group. But right now, a lot of our people aren't coming out of having spent a lot of the time, on ventilators and so on, right? So.
Yeah, I think another practicality about this is that the more different groups you have, in order to get statistical significance, you've got to get more people, and it requires more money. So we're sort of on a shoestring at the moment, and trying to prioritize our use of funds and participants in order to get good pilot data that will help us to get a big grant. I mean, ultimately, what we really do want to do is get as many people tested as possible. There's a lot of different factors that we would like to look at in addition to the measurements we're making. I mean, lifestyle factors, people's, maybe their cardiovascular risk factors, and we'd like to be able to see if there is any sort of relationship with those types of factors in addition to in relation to the measurements that we're making.
Great, thank you so much. Are you controlling for other factors like long-term exposure to air pollution?
We are asking questions about smoking history, for example, and because we do know where people live, if there were folks who wanted to get involved who are interested in sort of that in health environment side of things, we would be really welcoming to bring those kinds of people on to be able to do the analyses, because we you know, if we know where people live, for example, their postal codes, you can get at least some more idea about the long-term exposures and some certain environmental exposures. If we know, the status of their work environment, that's something that could all be done after the fact. But we'd also welcome, people who are experts in that field and the environmental neuroscience side of things to advise us on exactly what we ought to be asking up front. To make sure that we're getting the right kind of answers. Having said that, because we are planning to do the 6-month follow-up, and because we are doing some of the testing on the cognitive and emotional side remotely, we can always be adding in additional questions later. So even if we're not asking them at the very beginning and at the intake, we can add them in later. So there's a lot of that sort of thing. If people think it would be of great interest, let us know, and we're happy to work with you on it.
Great, thank you, Michael. Catherine asks, through contact tracing, could you perhaps enroll people who are not yet infectious early on so as to get a baseline in at least some of the patients?
That's a great idea. I like that.
Okay, great.
Of course, we're not quite at the contact tracing state yet, but as technology comes online, if we can harness that, I think it would be quite strategic.
Great.
I think what we were planning to do was a more scattershot approach, which is assuming that some of the people who came in initially as non-COVID or COVID negative would later have it, and then we would have their baseline. But this is a much more strategic way of trying to get those folks in. So thank you.
Yeah, great thoughts. Jen Reed says, will you be collecting blood samples as part of your study to look for biomarkers related to risk of neurological damage?
Yes, we will. So it's not actually a main focus of our research team, but we're collecting the blood samples, and we're going to be reaching out to others that are interested in that kind of biomarker and fluid biomarker analysis. And we'll collaborate with them.
Great.
Yeah. And that's one of the challenges we're facing in trying to put together an idea for the mobile testing unit. We actually have our eye on a mobile MRI system that we could sort of drive around from place to place. And we also want to be able to have, a full lab where we can be taking not just electrophysiological measures and behavioral measures, but also do biological samples of saliva, of blood, and so on, and then store all of that in a big van that we're driving across the country. So, and then delivering it for analysis to all the labs. So the logistics of that are a little bit complicated, but it's a very exciting idea. And then the idea also would be that the samples that we're collecting there can be used in a whole bunch of other studies that are looking at the other aspects of the virology.
That's super neat. Road trip. One last question as we're at time. Are you tracking the medical recovery period following COVID-19? Anecdotal, some people who had even minor symptoms like fatigue, coughing, light fever takes up quite some time to get fully back on doing sports and other recreational activities.
Yeah, so that's a very important thing for us to track. So you can have physical fatigue after you're recovering from the virus. But there's also the possibility that this is mental fatigue. There are certain parts of the brain that govern energization, and your ability to just get up to do stuff. So that is something that we're quite interested in tracking and seeing if that correlates with our measures.
Yeah, and even things like the sensory deficits. My brother-in-law is an ENT down in New Jersey. And so as you can imagine, almost everyone he knows, including himself, has already had this disease and has recovered from it. And even the smell and taste symptoms, some of them, the anecdotally anyway, they're lasting months, the loss of taste and smell, but it becomes more and more specific. So it's not in general, it's just smell of specific things or taste of specific things, which makes you think, again, it's not sort of a broader physiological mechanism. It's something actually that's happening in terms of processing in the brain. So, but it's really, that's something, again, with a 6-month follow-up and then hopefully longer follow-ups, we can be tracking all of that.
Well, this all sounds absolutely fascinating, and I look forward to the results of your study and that mobile lab testing around Canada. Thank you so much for joining us, everyone, and thank you, Dr. Sekuler and Dr. Graham. Thank you.
Thank you for having us.
Have a good afternoon.
Thanks. Bye-bye.